A lot of you know me. I met a few new people. I've been in the field of FA research for 12 years now. Sarah Vicente Muniz from my lab actually presented yesterday the scientific talk, and I know a lot of you guys have heard my sort of scientific spiel, so I kind of wanted to go things a little differently. So I'm going to veer off course a little bit. After I had sent FCF my talk, I thought, and nobody wants to listen to me talk about the science forever except for Evan, because he is nice and he'll tell me he wants to listen, he wants to hear it. But even he has heard it several times. So I'm going to try to go quickly through the slides because I have a couple of things that I think are going to be a little bit more interesting and a little bit more fun. And I don't know how it's going to play out, which actually makes things a little, it could backfire. So we'll see what happens with the cups, but I think that's part of the fun. I study metabolism, and for any of you who have had to take, I think it's like sophomore year biochemistry in like high school even, where you have to talk about all of these different metabolic pathways. And back when I was in school, and we really didn't have the computer use and the internet use, we had to write down and map all of these pathways by hand. And so we still torture students like Anne by making them do that because it's fun. But we don't have to do that anymore, because the nice thing about metabolism is that most of these pathways are really well known. How they interconnect are really well known now. It's been extensively studied. And so we can start to really trace what's happening in the body. So it makes it really nice. The other really interesting thing is that so many of you have probably been involved in things relating to genomics and proteomics, right? Talking about genes, talking about proteins. You guys hear a lot about the Fanconi anemia DNA repair pathway, where you have all of these proteins who have to come together and work together for the repair to happen properly. We're looking at it differently in that we're actually seeing in real time. I mean, and I can tell you, and even from our study, and that's really sort of part of what we're talking about today and why you have the cups in front of you. Your metabolism starts long before food ever even sometimes touches your tongue. So your metabolism is activated and starts before food ever gets to your stomach. There are so many sensors and neural transfers that are happening that are telling your body what it thinks it should do before the food ever actually makes it into your intestines, where the real fun starts to happen. And so it is real time. And, you're going to eat, depending on what you eat. Our study is on taking in carbohydrates versus proteins and fats and how that impacts someone with Fanconi anemia. But no matter what you eat, every single person in here is going to respond slightly differently to that. That doesn't matter if you have Fanconi anemia or not, right? That's also dependent upon whether or not you're 60 or 16, you're male or female, right? Whether you're underweight or overweight, right? Whether you exercise regularly or you don't. So metabolism becomes pretty complex because it's not just about the genetics, right? It's also about environmental factors and stimuli. I like to always sort of put this up there because I say, you are what you eat. I remember a few years ago, I think maybe it was the first year that I met Jasmine, actually, where she told me the story about how, I don't know if you remember, where you would wake up in the middle of the night and you would need pasta. I was so struck by that, and I get a lot of my research ideas by having these conversations and talking through sort of what that means. And that really just struck me, right? And we think about carbohydrates. A lot of people think about carbohydrates, sugar, and I need a candy bar. I need something sweet. That's not necessarily always true, right? Sometimes we can crave things like pastas, right? Sometimes we can, you hear a lot of really sort of crazy things happening when women are pregnant in terms of things that they may crave or things that they may not like to have. And the same happens across the board for everybody, right? And so sometimes you can smell something and it instinctively is like, I need, I don't know, a steak. I love steak, so I need a steak, right? Or something like that. You're definitely very much what you eat. Your body is constantly breaking things down to build things back up, right? Breaking things down to build things back up. It's a constant cycle. You don't know it's happening, thankfully, but it's constantly working on everything that you're taking in. It can handle some things that you take in much more easily than others, right? Simple sugars are really easy for the body to break down and use as what we call quick energy, right? That's the ATP that you see. That's sort of the fuel that the body's wanting to take in. And you can use glucose and simple sugars pretty readily. I mean, especially if you're just, I like to, which is completely counterproductive, and I'm going to admit that. But when I have Frosted Flakes, it's like a little bit of dabble of like sugar across the top because it just makes it taste a little bit better. Don't look at me like I'm crazy. I'm from Ohio, okay? So, or grapefruit, right? Like I grew up eating where we would cut a grapefruit in half, and then my grandpa and I would sprinkle sugar across the top, and that was the best thing ever, right? So that's really easy for our bodies to break down and to use. Whereas if we're taking in the same thing, carbohydrates, but in a more complex form, right? We're talking about some of the things that we have up here we're going to talk about, like the whole wheat, right? Or if we're taking it in combination, right, with high protein, healthier fats, our body is going to process things differently, and it's going to take a little bit more time for it to break it down. I do metabolism, and we use, and I was joking earlier, which unfortunately is not a joke, but we look at every type of biological fluid, right? And so we look at blood and urine and saliva fairly regularly, but we also study feces, right? Because we can get a lot of information from microbial gut bacteria, right? We study, a lot of them are doing tissue cultures, right? Tumor tissues versus normal tissues to study the metabolism of different cancer types. And so all of those types of things we can process. We put it into a large instrument, and so that's the NMR analysis, and it gives us this, by any other means, it's just all of these dots and lines. And so those are all representative of different metabolites. And it's ones you've heard of, right? So it's glucose, first of all, right? You can see a number of sugars. Right? You can see your branched chain amino acids, if you remember those. You can see lactate, right? A lot of people talk about lactate, alanine. You see in a lot of the health food things like tryptophan, right? We pick that up on the instrument. We also see any sort of remnants of antibiotics or drugs, sort of exogenous components, right? Those all come up as a series of peaks, and then we know how much is in sort of a given blood sample or urine sample, and that tells us whether or not it's in a normal level, or maybe it's low, or maybe it's high. And then we can start to map it back to those pathways that people long before me sort of worked through and really know about. Most of you at this point know about the study that we did a few years ago, and Sarah presented on the data. But we sort of did just that. I'm sure, I don't know how many of you have been told, that in terms of like BMI, maybe for years, the only thing that was used as a measure of whether or not you were overweight or underweight was your BMI. I also had the same thing happen to me. And as a very active athlete, I was always overweight. I was in the morbidly obese category by BMI most of my childhood and most of my life. Even as a collegiate athlete, I was designated as morbidly obese by BMI standards, right? So we know that it's not the best way to look at the composition of an individual. So we took it much further and started looking at multiple sort of nutritional aspects. We did grip strength tests, right? We talk about, especially in males with Fanconi anemia, who are trying to build muscle mass. It's very difficult to do. So we talked about sort of what is the grip strength like? Do they have any grip strength? So we looked at also an in-body analyzer, right? So you basically stand on this oversized scale and you're holding these little handles. And so it's going to send sort of electrical current through, and that's measuring things like where do you hold fat, right? That's really important. If we're trying to put weight on, but we're only able to get you to gain weight in the gut, right? We know that that has things like cardiovascular risk associated with it, right? So we want to make sure that we're actually building muscle. We want to make sure that we're actually putting on good fat as opposed to just fat, visceral fat, or fat in the trunk region. So we looked at all of these different aspects. I don't want to waste a ton of time on this data. Most of you have seen this data. So we did indirect calorimetry, and that's actually a pretty cool thing. So it's tented breathing. You basically just lay there everybody basically took a nap. It's like white noise inside of this tent. And you lay there. And so you're just, all we're doing is monitoring oxygen in and carbon dioxide out. The first thing that we noticed was that you really should, we have you drink glucose. So it was actually about the amount that you see, in the cups. It was 50 milliliters. So it was a little amount. And it was 25 grams of glucose. So think about the amount of sugar in about 2/3 of a Snickers bar, right? So I don't know who eats 2/3 of a Snickers bar, right? You're going to eat the whole bar, right? But 2/3 of a Snickers bar. And so it's not a ton of sugar. It's definitely not as much sugar as you're going to take in and say like a bowl of cereal in the morning. And for the controls who do not have Fanconi anemia, they responded as we suspected. They had almost no response, really. I don't even know if I can use a, yeah. So this is what we expect. They should have almost no response, but they're in a fasted state, right? So we had them fast. Everybody in the study fasted, and then they are going to take in just a small amount of glucose. Typically, that would trigger your metabolism to kick in just a little bit. Not a lot, because it's a tiny amount, and they're a typically sized person, right? But we see a tiny little blip. Then, when we come to the individuals that have Fanconi, now again, they're smaller, right, in body size overall, so they have a lower resting metabolic rate. That's not unexpected. But what happened within 30 minutes of them taking in just that small amount of glucose really was pretty shocking. Immediately, their resting metabolic rate, as it was calculated, right? So first of all, we noted that they started taking in the oxygen consumption was reduced, and the carbon dioxide they expelled was reduced. So it was sort of shutting down that part of metabolism just by taking in a tiny amount of glucose, right? And then that persisted. We hadn't been measuring they were really not, very interesting to look at. So we had only measured the controls out to 30 minutes, and we did a 180 at the end of the study. We thought we need to see what's happening with individuals with FA prolonged, and even at 4 hours after just taking in that tiny amount of glucose, they still had not metabolically recovered back to baseline. So taking in that sugar was having a profound impact on their body metabolically. So we really started to try to look and see what was happening. And most of you have seen this data before. So this is just, here we're measuring, we're using our instrument, and we're looking at, this is just the glucose that was already in the body, right? So this glucose isn't labeled. The difference was that I'm actually able to modify the carbon atoms on glucose, and so now I can pick it up and trace it in my instrument. So when we gave them the glucose, I can actually trace that glucose. So I can tell the difference between the glucose that was residual glucose that was still in their body after fasting and the glucose that we had now given them. And now we're looking at what are they doing with that glucose? And we thought, well, immediately, and I was completely wrong. I thought they can't be using it, right? We thought it's probably in the urine, right? Because their metabolism is shutting down, so to speak, by, the bionutritionist said, well, this means they're just shutting down their metabolism if they're not, sort of expelling the carbon dioxide. It's really quite the opposite. What we saw happen was a rapid glycolytic rate. So you can see for the individual, so if it says 13C, that's where we've modified the carbon. So now we know that this is and has to be the carbon that from the sugar that they drank. So this is the sugar that they drank. So you can see this is the raw sugar here in panel A. So that's the raw sugar. So we know that, first of all, you can see pretty clearly that in the controls, it sort of peaks, as it should, not very high, and then it just sort of goes back down. As I mentioned, it's not very much sugar. But the concerning part was that it just kept climbing in the individuals with FA, and it didn't go back to baseline in the 4-hour period. 25, a tiny amount of sugar, right? We just kept following them. And it climbs to a really high level. This is in the blood. And we thought, well, maybe it's just sitting in there, right? Maybe the blood, or maybe the glucose is just in the blood and it's not being used. Completely the opposite. So if you think back to the pathways, and I said they've been mapped well, one of the things that happens with glucose, we talk about glycolysis, it rapidly is going to convert it to things like pyruvate, lactate, alanine, and we see just that. So you see the exact same trends. So they're rapidly using. It's not that they're not able to use and process the glucose. They're using it very rapidly. We talked about how it's an easy way to make ATP. Well, that would be the goal. I mean, that's really that's what the aim is, right? And so they're turning this over very quickly. So the other thing that we really wanted to look at was, okay, what's happening? I've always sort of said that individuals with FA, especially those who are underweight and can't maintain weight, are in this sort of constant state of starvation mode. Like their bodies believe that they're in that state of starvation. And when that happens for, say, someone like me, where I've gone, let's say, 24 hours without food, then I'll go into ketosis, right? I'll run out of glucose and I'll start using fat stores, right? I'll start burning fat. That's a natural process. Well, in individuals with FA, we actually see that phenomenon within 3 to 4 hours after finishing a meal. So it's a very quick and very abrupt turnaround to the need to use fats and not recognize that they have the sugars and that they have the glucose that their body needs. The other thing is that even at baseline, these are the ketone bodies, right? So this means that this is just 8 hours of fasting. These are incredibly high levels of all of the ketones, which means that after 8 hours just going to bed and waking up, all of the individuals with FA were in a really significant state of ketosis, right? And a lot will, have noted to me in the past of waking up nauseous, waking up just not feeling well. You're in a significant, right, ketotic state by that point in time. And so it's not surprising that you maybe aren't hungry because you need to eat something, but you don't feel really well in terms of consuming food. So it's not surprising to see this. So the other thing that we looked at was actually hormone influences, hormone levels. And so all of the individuals, important note to make, I think, is that all of the individuals in my study were post-pubertal. And so we're now, Ming Wong from Cornell, who I'm working with now, we're getting ready to roll out a clinical intervention trial in individuals with FA ranging from 2 to 8, because I think it's really important to see what happens, right, in that younger population. Are we seeing the same phenomenon? Because we know that hormones are going to play a large role in terms of how our bodies recognize things. And several people in Oinika's did the overview, and several people noted changes in sort of hormone levels regulating things. I'm going to remind you again, the clinicians and me are like, well, what's happening with insulin, obviously, right? So an exclusion criteria for our study was any sort of diagnosis of, medically diagnosed insulin resistance, right? To do like obviously diabetes. All of these were, no one could be on any medications that may sort of be known to regulate glucose. So we felt like we had no one in the study who had never been clinically diagnosed. And you're talking about individuals with FA who some were upwards of 30 years old. So they had been through a number of clinical diagnoses. What we see is really no insulin response. So you remember, if you see, that's a high level of glucose, but the body is not recognizing in terms of the pancreatic beta cells, which should be, it's not recognizing that glucose is there. But what we do see is that things like, I used to always say that if obesity researchers really paid attention, then they would realize that they have a model of basically the diet that they are attempting to create in an individual who has obesity in an individual who has FA. So if you think about the Ozempic that's everywhere, the semaglutide, the GLP-1, right? If you're thinking about these sorts of things, when within 2 hours of taking in a small amount of glucose, you see a huge secretion of active GLP-1 in individuals with Fanconi anemia. Now, GLP-1 is secreted, right? It's a gut-derived hormone. So that means that it's recognizing and it's secreting it into the bloodstream. And this, again, it persists throughout the entire study. And so they're certainly having a response, right? They're just We talk about how lack of appetite, right? We talk about the feeling of fullness, right? Things like GLP-1, ghrelin, other gut-derived hormones, those are what regulates whether or not you feel full, whether or not you feel like you need to eat. Well, if you have elevated GLP-1, but you really they didn't have any food in there. I mean, they were hungry. They were getting hangry. I mean, at this point, they were not happy with me, right? But so they knew that they needed to eat. Their stomachs were making noise, right? But if you asked them if they had an appetite and they felt hunger, that's a different question. Two things are happening. And again, so this is just another look at other hormones. We really did not see that response that we really should have seen in those insulin-related hormones. We looked at a lot of other hormones, and we've been mapping that sort of in terms of how that relates to. We have a lot of studies at looking at liver issues and liver failure at Cincinnati Children's as well. So we've been mapping that to sort of the different organ systems, right? Because that's the other thing, is that we're able to do studies in cell culture, and we're able to do studies in some mice models, obviously. But when you're talking about a study where you're looking at metabolism in an individual, metabolism happens differently in every organ system, right? And so some organ systems aren't even able to process certain metabolites. So we have to bring all of that information together to see how it makes sense for the body. There was a really important note that was made yesterday, and I'm going to say, is it Holzmakers? Han Holzmakers? He's new to this was his first time at the FA Field. I don't know if any of you actually saw his presentation. I think you guys were in here. It was quite incredible. He's studying another even rarer genetic condition, and they did a dietary modification and were able to basically go from a child who had tremors, who could not walk or function, to now a child who is walking and writing and reading. It's incredible. And so he made a very important point, and he and I have talked at various times, and I think we are in agreement. A typical person obviously has all of these things happen, right? They have to regulate circadian rhythm. They have DNA damage, right, and repair that's happening. They're going to have inflammation from time to time, but they're not having to regulate all of these processes simultaneously at the severity of an individual that has a DNA repair defect, such as Fanconi anemia, or those that he's studying, right? So we think about the fact that we have what we need. All of these pathways are regulated by things that we take in from the diet and our vitamin supplements. So what we think is happening is that our bodies are having to almost pick and choose. Well, I'm not deficient in this, I'm not deficient in this, right? It's such a fluid process. It's very difficult to accurately measure without doing these tracers. It's really difficult because you're just getting a snapshot, right? So a level could be okay, and then it's right, and then another process needs it, right, for a key repair. We have to remember that a lot of these regulators, and they're called cofactors, so they're actually required. They have to bind to the proteins to activate them and then allow for all of these processes to happen. Those are all coming from essential nutrients. And so if our body has to say, I really need, right, we talk about survival, right? That's what he talked about. Well, we're going to pick attempting to repair the DNA, right, every day. But that means that things like, I've spoken with a lot of you about switches and disruptions in circadian rhythm in your sleep cycles. That's pretty common. We talk about, obviously, we know inflammation. We know mitochondrial dysfunction is a thing, and metabolism. So you're attempting to do all of these things. Your body's working so hard all the time. You need more, right? And so that's why kind of like what we're talking about today, right? We need more. We have to approach it differently. We have to think about it a little bit differently. It doesn't mean that we have to, sort of, I think, turn things upside down, right? That's not feasible, right? So that's why, I went to the store yesterday here and just sort of picked up things that I was looking at where it's like you're eating goldfish, you're popping goldfish now as a snack. That certainly your body's not handling that well, right? So let's think about, right, even if it's just a snack in the middle of the day, let's think about something that we know that where we can be giving a little bit more or a little bit something else to the body to just hold it over, right? Especially before we go to sleep. And we'll talk about that a little bit. Especially before we're going to go through an 8-hour fast where we know that our body is going to spend, we're sleeping, but your bodies are working incredibly hard and they're still doing all of those things, right? So that's why you're waking up, either in the middle of the night, because metabolically speaking, your body is running on all cylinders. So you probably are hungry, right? You probably have ran through all of your food stores, right? So that's a real thing that's actually happening. It's not, when I hear someone say, that's not real, right? That is real, right? Your body is right. Your body is hungry because it's working so hard. And I want to get to the good stuff. So, and I'm not even going to go through this. You guys know this. I can talk till I'm blue in the face. I've talked to some of you, and you're going to do what you want to do. I'm going to do what I want to do, right? This is real life, right? It also has to be feasible in terms of whether or not, right, you're working, right, or you're going to class, right? You're raising a family, right? And so I'll be honest, how many times are you like, well, fruit snacks for dinner, right? Because you're eating what, right? You're eating what the kids eat, right? So it has to be manageable, and it's not going to be every day, but there's just little things that we can talk about, that we can do a little bit differently. I want to do this before we have to shut down. And then you guys can sort of taste and look at this food. This is food that I've got. So in front of you, if you don't have cups and you want to participate, there are extra cups that are numbered. They're numbered 1 through 5. Okay, so I want you guys to start thinking, and if you want water, you can go get water. We're not gonna do X Factor taste. You can't have any water to drink. So I want you to think about sort of how you're tasting, and I don't know the answer. I'll be honest, I don't know the answer. But I want you to think about, and you can do this in the next session, right? It doesn't have to be immediate. But smell it. Some of them, I don't know, maybe they'll all smell to you, maybe none of them will smell to you, right? But smell plays such a huge part in terms of how we taste food, right? So these are the 5 main tastes, right? You have sweet, savory, bitter, salty, and different parts of your tongue are going to taste the food differently, right? Now, if you had a transplant, then that's going to be another confounding variable, because that could be something that's happened completely different. I just want you to think about it. Taste it with your nose plant, right? With no smell. See if there's anything. See what happens when you release and you have that taste. Take another taste. Take another taste, then with the ability to smell it. See if. Don't lie about it. If you can't taste it, then just say that you can't taste it. All of the tastes are represented. So if you're like, there isn't one that's bitter here, there is, but be honest. Okay, and there's extra papers if anyone needs paper. Think about how many times do you think that you have to consume a new food before you know whether or not you like it? Andy. How many times do you think you need to consume a new food before you can say that you like it? A lot. You have to consume a new food up to 20 times before you can say whether or not you like it. So when you taste something once and you say, I don't like this, think about that whenever you're thinking about introducing new foods, right? Berries, right? If you're thinking about fruits, berries are a wonderful source, right? We talk about antioxidants all the time, right? Berries are a wonderful source of antioxidant. So here are 3 types of berries that are a great source. Like if you're thinking, I don't know. And I'm putting these out here because it's possible that you've seen these at your store and you're like, I'm not going to waste money on them. I don't know if I like it or not. Just taste it. And I've got a bunch of cups. These are actually, I don't know how I feel about them. But I'm using these as an example. You've probably seen the naked drinks. So they have a fair amount of sugar in them. They have a regular and they have a low sugar option. So, and again, we have little cups up here. I want you guys to take the opportunity to taste them. So I picked Green Machine. I really like Green Machine because, again, green vegetables, right? Introduction of green vegetables, right? So berries, green vegetables, right? Those are great sources of nutrients. These are no sugar added drinks, right? So it's no artificial sugars added. So they are sweet, but they are because they actually have fruit in them. Like it's the actual fruit that's in them. So you can go a little bit less in terms of the sugar if you go like with the green machines, and this is glorious greens, because they have a little bit more in the way of vegetables in them. But I want you guys to taste this. I know people are looking crazy. So these are the types of things that I don't want you going forever and not eating. I know you're busy, but all of these things don't require refrigeration. You can take these with you. Nuts are an amazing source, right? So I have pistachios. Again, I'm going to be perfectly honest with you. I was 21 years old when I had broccoli for the first time. I was 32 when I had a Brussels sprout for the first time. I'm not blaming you for this. I grew up in Appalachia on meat and potatoes, okay? I thought corn was a vegetable. So this is not on you. Like this is just real life, right? So again, these are actually pretty good. So you can still have your taco, eat the taco. Like don't not eat the taco, please eat the taco. But these are actually really good. So these are high protein, carb savvy wraps, right? And there's a ton of different options, a ton of different types. There are spinach ones, right? But and I put this up here. I'll be honest. I didn't open this. I hate tuna. If you guys want to open it, I will scour at you and you can try it. But this is actually a nice one because they actually make them in different oils. So this is in sunflower oil. So there's a lot of additional, when you look at the back and compare this to the other tunas, this one has a lot of extra really good fats in it, right? So those are the types of things that we can look at. And then I will say, do not just go and buy. This says keto. I hate the use of the term keto, but this is actually a pretty decent one. So it's bitter, like dark chocolates, which dark chocolates are a wonderful option, right? So I'm assuming at this point some of you have made it through some of the drinks, and there were questions about what cup number 2 was. I did not intentionally make it cup number 2, but there were jokes, but. Right, bitter chocolates are actually a wonderful, wonderful source, right? Whenever you're consuming them. And so this one is a nice one because it's a combination of nuts and chocolates. And then I like these, and these are good for travel. And so these are the Kind bars, the Kind mini bars. So I find that the Kind bars actually do a good job. You have to watch because some of them are like fruits and berries and caramel, and those ones are definitely high in sugar. But they have ones that are dark chocolate, nuts, and sea salt ones, and those ones are actually they're really nice in terms of, and I will say. Right, so for you guys, keto is also about like lower calories and higher fats. That's not what we're trying to do, right? It's being more conscious, right? So I bought this, and you're welcome to try it. So some of the parents at this year at the Painted Turtle, we were talking about it, and they switched over. So Dave's Breads is actually like a keto bread, but it's the same idea where it's bread that has like nuts and grains in it. Right, so it's not just that the white bread is like, there's like nothing to it, right? It's just sugar, sugar, sugar. So when you're thinking about bread, right, please don't stop eating bread because bread is amazing. But think about it in a way of like, let's get bread that's just a little denser, right? You just want nutrient dense as opposed to other things. And then this is granola. So when you talk about things with almond or like coconut oils and coconut milks and things. So those are just a really easy way to add healthy fat. So I want you guys to think about that, and I have some knives. Again, it doesn't take a lot, right? I mean, I know it feels like that, but if you already eat eggs in the morning, maybe throw on a little bit of cheese, right? Right. So something that's a really good source of protein. Eggs already are a great source of protein, right? If you're going to do bread and you like bread, then by all means, maybe consider an avocado. Avocados are a great source of protein and fats, right? So I think don't try to completely recreate everything that you're doing. If you can pick one thing, and we know, don't go forever, right? I know it's so easy to get sort of occupied and to skip breakfast or lunch or dinner. I will say, never skip dinner. If you're an individual that has Fanconi anemia, never skip dinner. Never skip a bedtime snack. I'm giving you, never skip a bedtime snack. I'm serious, never skip a bedtime snack. If you can have a scoop of peanut butter and some crackers, like, wonderful, right? Something that's like, that's gonna sit, what is, like, I don't know what my parents used to say, like, that's gonna like stick to your bones, right? Like, something that's gonna stick to you, right? Because that means that it's gonna take the body longer to break it down, and that means that it's gonna sort of not trick because the food's there, but that means you have longer time before your body goes into that fasted state, right? So that's what we're trying to achieve overnight. So if you're struggling with weight maintenance and with weight loss and weight gain, think about going heavier in your evening meals and evening snacks, right? Because we mentioned, one thing that we're looking at now is that shift in circadian rhythm. So a lot of times what the circadian experts will say will be, eat later. Because that means that your metabolism is probably ramping up. And if you're a heavy breakfast lunch eater, you've ran through the bulk of your energy stores by bedtime, right? So you are, right? You're hungry. You're just waking up hungry. That's absolutely possible. So think about sort of extending, and then go with the idea of, snacking. Oh, I didn't even introduce this, but I want everybody to try it. I'm going to try it. So I found this on the shelf. It's called Fit Butters, but it's not meant to be, obviously, I'm assuming that like weightlifters or bodybuilders use it, but it has whey protein in it, but it's cashew butter, and this one is chocolate chip cookie dough. Yeah. So it's only 10 grams of carbs. So it's actually pretty low in terms of carbs. So they have all different, and I've seen different ones. They have different flavors, but these ones are specifically, it's added whey protein, and they're made with coconut oils. These are, and we were talking earlier. Yeah.
How does this all work when they tell you have to fast like 12 hours before surgery? It doesn't, right? You fast because of surgery, right? You've got to fast, right? If you've been in the hospital a lot and you've fasted a lot, there's no way that hasn't had an impact, right, on you in terms of your metabolism. There's no way that it hasn't shifted. If you spend a lot of the time in the hospital and obviously you're not getting sleep and your sleep-wake cycles are all thrown off, there's no way that that's not impacting your circadian rhythm, right? I think that we try to minimize it as much as possible, but the reality is that, there are procedures that require you to fast, right? And you can try to be conscious and tell them, right, we call it fasting intolerance. I'm fasting intolerant, so can we minimize the amount of time that I need to fast? Can we make sure that we're ready to go with the blood draws so that I'm not fasting for any more time than I absolutely have to? But yeah, we call that fasting intolerance. And if you're just upfront and like, listen, I would prefer to only have to fast for this amount of time, please, like, do your best to make sure that you've got that nurse in there to do these blood draws and this procedure on time. Because I don't do well if I've got to fast. Yeah, so I just, of course, I ended with this because I couldn't start with it. So I do all of this work for all of you, but especially for him. And so he unfortunately, left us recently, and so I just want to say that this work and all the rest of the work that I do is dedicated to him and to all of you. Thank you.